Welcome you all, dear students. In this module, I, Dr. Shushma, we will be talking about metabolism of carbohydrates under aerobic conditions. In these sections, you will be learning about how the metabolism of carbohydrates under aerobic conditions occurs. In general, metabolism refers to how the body provides the energy that is necessary for its work and its during exercise. To speak about a general introduction, generally energy metabolism is defined as the biochemical reactions which takes place in the body that involves changing energy from one form to another or in transferring energy from one chemical substance to another. There are two forms of energy metabolism coined: catabolism and anabolism. Catabolism includes the chemical reactions in which larger molecules are broken down to smaller ones, and anabolism includes the reactions in which larger molecules are formed from smaller molecules. In catabolic reactions, chemical energy is made available as the larger, higher energy molecules are broken down. In anabolic reactions, energy must be added so the smaller, lower energy molecules can form the higher energy, larger molecules. It's important to note that an excess consumption of any or all of this food source, including carbohydrates, fats or proteins, doesn't result in more energy being produced. Rather, it results in the consumed excess being converted to and stored as adipose tissue. There are three separate energy systems through which ATPs can be synthesized. These are as follows: ATP-PC system, which is also known as the phosphagen system; anaerobic glycolytic system, which is also known as lactate system; aerobic system, which is also known as slow glycolysis or oxidative phosphorylasis. The ATP-PC system and anaerobic glycolytic system are both anaerobic systems, meaning that oxygen is not used by this system to synthesize ATP. The aerobic system, on the other hand, relies heavily on the oxygen to synthesize ATP. Most exercise involves ATP being synthesized through a mix of all three systems. The factors that determine which system is most dominant at any time are the intensity and duration of exercise. The diagram below is an example of the percentage contribution of energy provided by each system during the initial 6 minutes of a run. When exercise begins, energy will come from the anaerobic energy systems. The initial 10 seconds or so are almost exclusively through the ATP-PC system. As exercise continues, the anaerobic systems become depleted due to the limited stores of ATP, PC and glycogen. And the aerobic system becomes increasingly dominant as it can break down more complex fuels for energy, such as fats and proteins as well as glycogen. The higher the intensity of the exercise, the quicker the anaerobic systems will be depleted. For exercise to continue once the anaerobic systems have become significantly depleted, the intensity of exercise needs to drop to a level that allows aerobic system to provide enough energy. So by end of this module, you will be understanding and learning about what is a metabolism, how the aerobic metabolism works, how the aerobic metabolism of carbohydrates works. Now let us learn how aerobic metabolism works Aerobic metabolism is a way for your cells to convert fat, carbohydrate and sometimes protein into energy. But only in the presence of oxygen, aerobic metabolism is slow, so it is useful for sustained activities like jogging or dancing rather than short bursts of effort like sprinting or weightlifting. Aerobic metabolism converts fat and carbohydrates into units of cellular energy called ATP. Aerobic metabolism is very efficient, producing 34 molecules of ATP for every molecule of glucose. Compared to the 2 ATP molecules anaerobic metabolism generates. Aerobic metabolism is also the only means by which your cells can use fat for fuel, which is part of the reason why aerobic activities like swimming or cycling are such an effective means to lose weight. As you know, your body performs aerobic metabolism all day long to provide energy for activities of daily living. Because aerobic metabolism requires water and oxygen, it is the reason that humans breathe constantly and part of the reason why you need to drink water. Carbohydrates are a necessary ingredient for aerobic metabolism, even to burn fat. Because the muscles and liver can only store a limited amount of carbohydrates between meals, aerobic metabolism is also the reason why you need to eat carbohydrates every day. Although your body can metabolize proteins for energy in the absence of carbohydrates, it is a slow and inefficient process. Aerobic metabolism in the absence of carbohydrate can only keep up with activities with a very low energy cost, leaving you feeling sluggish. Aerobic metabolism provides carbon dioxide and water as a byproducts. The extra carbon dioxide production from keeping up with the demands of exercise is what makes you breathe hard when you exercise. Humans can only use 75% of the oxygen they inhale at their maximum exercise capacity, meaning that there is no shortage of air coming in. Instead, it is the extra carbon dioxide built up in the blood and the need to expel it that makes you short of breath. An overview of carbohydrate metabolism. Students, by now you have understood about how aerobic metabolism works in a system. Now you will be studying aerobic metabolism by using carbohydrates as a fuel. Carbohydrate metabolism denotes the various biochemical processes responsible for the formation, breakdown and interconversion of carbohydrates in living organisms. The most important carbohydrate is glucose, a simple sugar which is called monosaccharide that is metabolized by nearly all known organisms. Carbohydrate metabolism begins with digestion in the small intestine, where monosaccharides are absorbed into the bloodstream. Blood sugar concentration are controlled by 3 hormones, namely insulin, glucagon, epinephrine. If the concentration of glucose in the blood is too high, insulin is secreted by the pancreas. Is required for the normal functioning of the organs in the body. Many tissues can also use fat or protein as an energy source, but others such as brain and red blood cells can only use glucose. Glucose is stored in the body as glycogen. The liver is an important storage site for glucose. Carbohydrate metabolism begins with digestion in the small intestine, where monosaccharides are absorbed into the bloodstream. blood sugar concentration are controlled by three hormones, namely insulin, glucagon and epinephrine. If the concentration of glucose in the blood is too high, insulin is secreted by the pancreas. Insulin stimulates the transfer of glucose into cells, especially in the liver and muscles. Although other organs are also able to metabolize glucose. In the liver and muscles, most of the glucose is changed into glycogen by the process of glycogenesis, that is also called as anabolism. Glycogen is stored in the liver and muscles until needed at some later time when glucose levels are low. If blood glucose levels are low, then epinephrine and glucagon hormones are secreted to stimulate the conversion of glycogen to glucose. This process is called as glycogenolysis, which is also referred as catabolism. If glucose is needed immediately upon entering the cells to supply energy, it begins the metabolic process called glycolysis under catabolism. The end products of glycolysis are pyruvic acid and ATP. Since glycolysis releases relatively little ATP, further reactions continue to convert pyruvic acid to acetyl-CoA and then citric acid in the citric acid cycle. The majority of the ATP is made from oxidation in the citric acid cycle in connection with the electron transport system. During strenuous muscular activity, pyruvic acid is converted into lactic acid rather than acetyl-CoA. During the resting period, the lactic acid is converted back to pyruvic acid. The pyruvic acid in turn is converted back to glucose by the process called gluconeogenesis under anabolism process. If the glucose is not needed at the moment, it is converted into glycogen by glycogenesis. You can remember those terms if you can think of genesis as the formation and beginning. The aerobic process of carbohydrates are given below: glycogenesis and gluconeogenesis. We'll now understand about what is glycogenesis. The synthesis of glycogen from glucose is known as glycogenesis. Glucose entrapped in the cell as glucose phosphate is mutated to glucose 1-phosphate by the enzyme called phosphoglucomutase, which in turn is attached to UTP by the enzyme glucose 1-phosphate uridyl transferase, forming UDP glucose. Glycogen synthase adds glucose to glycogen primer with a few glucose units by making alpha 1 to 4 glycosidic linkages, and thus forms a linear chain of 10 to 12 glucose residues, all linked by alpha 1,4 glycosidic linkages. At this time, another enzyme, that is branching enzyme, removes 6 to 7 glucose units from the linear chain and transfers them to the other chain and attaches by alpha 1,6 linkage, therefore creating a branching point. The process of addition of glucose by glycogen synthase to the linear chain and branching enzyme creating the branching points is repeated, and thus glycogenesis is completed. Now we'll understand about gluconeogenesis. Gluconeogenesis is formation of glucose from non-carbohydrate sources. Gluconeogenesis helps to maintain the glucose level in the blood so that the brain, RBC and muscle can extract glucose from it to meet their metabolic demands when dietary glucose is low. This process is very much necessary in the body because brain and RBC utilizes only glucose as energy fuel. The major non-carbohydrate precursors of glucose are lactate, glucogenic amino acids and glycerol. Lactate is formed by RBC in glycolysis because mitochondria are absent. Lactate is also formed by active skeletal muscle when the rate of glycolysis exceeds the rate of TCA cycle. The pyruvate formed is converted to lactate. Amino acids are derived from proteins in the diet and during starvation from the breakdown of proteins in skeletal muscles. Glycerol is derived from the hydrolysis of triglycerols. Gluconeogenesis occurs mainly in the liver and kidney. It also occurs in brain and muscles to some extent. Gluconeogenesis occurs during starvations to clear lactate formed in RBC and muscle when carbohydrates in the diets are low during pregnancy, during lactations and febrile disease. Gluconeogenesis is almost the reversal of glycolysis, expecting at 3 steps which are irreversible in glycolysis. These steps are reversed by enzymes known as the key enzymes of gluconeogenesis, that is, those enzymes specific for gluconeogenesis. The main key enzymes of gluconeogenesis are pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 diphosphatase, glucose 6 phosphatase. Now we'll understand about the role of 2,6 biophosphate in gluconeogenesis. Fructose 2,6 biphosphate is a metabolite which is allosterically affects the activity of enzymes phosphofructinase 1 and fructose 1,6 to regulate glycolysis and gluconeogenesis. Fructose 2,6 biphosphate is synthesized and broken down by bifunctional enzymes called phosphofructokinase to fructose 2,6 biphosphatase. The synthesis of fructose 2,6 biphosphate is performed through the phosphorylation of fructose 6-phosphate using ATP by the PFK2 portion of the enzyme. The breakdown of fructose 2,6 biphosphate is caused by dephosphorylation catalyzed by FBPase2 to produce fructose 6-phosphate and pi fructose 2,6 biphosphate, which stimulates glucose breakdown. Further, through reduction of gluconeogenesis through allosteric inhibition of fructose 1,6 biphosphatase. Now we'll study about aerobic catabolism of carbohydrates. Glucose can be broken down to acetyl-CoA. In fact, the reactions by which this happens are the same as those of anaerobic glycolysis, except for the final one. In both anaerobic and aerobic glycolysis, one glucose molecule is converted to 2 pyruvic acid molecules. Incidentally, biochemists don't like the term aerobic glycolysis, but physiologists often use this term. In anaerobic glycolysis, each pyruvic acid is converted to a lactic acid molecules in the final reactions. In aerobic catabolism of glucose, each pyruvic acid molecule is instead converted to an acetyl-CoA molecule in a single reaction, besides making acetyl-CoA for the Krebs cycle. This reaction also generates an NADH2 for the ETS, that is electron transport system. In addition, a carbon dioxide molecule is formed for each 3-carbon pyruvic acid that is converted to a 2-carbon acetyl CoA. There are two important aspects of aerobic glycolysis. In certain reactions of pathway, ATP are formed, a net production of 2 to 3 per glucose molecule. Secondly, an additional 2 NADH2 molecules are formed and the electrons from this NADH2 molecules enter the mitochondria to be processed by the ETS system. In aerobic glycolysis, glucose from the blood or from stored glycogen is broken down to acetyl CoA and NADH2 and ATP are produced. Carbon dioxide is also produced. No oxygen is used in these reactions of glycolysis, but the reactions that convert glucose to acetyl CoA are dependent on the Krebs cycle being able to accept the acetyl CoA, and this is in turn is dependent on oxygen consumption in the ETS system. This makes this pathway of carbohydrate breakdown part of aerobic metabolism. ATP can be generated by aerobic metabolism using energy ultimately derived from carbohydrates, proteins or fats. Most of the ATPs is formed in the electron transport system by the process known as oxidative phosphorylation. Some is also formed in the Krebs cycle and in glycolysis. Electrons are brought to the ETS by NADH2 and FADH2. Then these electrons are transferred from one substance to another in a chain of reactions until they are ultimately transferred to oxygen. This converts the oxygen to water, and in this process energy is made available to phosphorylate ADP, forming ATP. The electrons brought to the ETS originally comes from the carbohydrates, fats and proteins. NAD is converted to NADH2 in glycolysis and in the conversion of pyruvic acid to acetyl CoA and in the Krebs cycle and in beta oxidation of fatty acids and in the conversion of lactic acid to pyruvic acid. FAD is converted to FADH2 in the Krebs cycles and in beta oxidation. The Krebs cycle requires acetyl CoA as an initial reactant. This acetyl CoA can come from breakdown of fatty acids, sugars and some amino acids. Products of the Krebs cycles are ATP via GTP, NADH2, FADH2 and CO2. Beta oxidation requires fatty acids as initial substrate. Products of beta oxidations are acetyl CoA, NADH2 and FADH2. Glycolysis requires sugars, especially glucose, as initial substrate. Products of aerobic glycolysis are ATP, NADH2 and pyruvic acid. The pyruvic acid is then converted to acetyl CoA with formation of NADH2 and carbon dioxide. After the amino acids have been removed, amino acids from proteins can be substrates for the Krebs cycle either directly or indirectly. End products of the aerobic breakdown of all the initial substrates are carbon dioxide and water. Carbon dioxide and water have very little energy stored in them. This means that almost every bit of energy is squeezed out of the original substrates. Much of this goes to ATP formations, some is converted to heat. Of the many chemical reactions that make up the aerobic metabolism, oxygen is used in only one, the final reaction of the ETS, in which hydrogens are added to the oxygen to form water. Without the final reactions, however, all the reactions of the aerobic metabolism would quickly stop. In other words, all the reactions of beta oxidation, aerobic glycolysis, the Krebs cycle and ETS ultimately depend on oxygen use in the ETS. That is why all of these reactions or pathways are included in aerobic metabolism. Dear students, now let us understand about the advantages and disadvantages of aerobic metabolism of carbohydrates. The main advantage of aerobic metabolism over the other two methods of forming ATP is its huge capacity for ATP formation, that is total amount of ATP that can be formed. This is to large extent due to the ability of aerobic metabolism to use carbohydrates, fats and proteins as the initial substrates. Carbohydrates provide the smallest store of energy. In a person average body size and on a normal mixed diet, a total about 2000 calories of energy is stored in its various forms: glycogen in muscles, glycogen in the liver and glucose in the blood. In contrast, the same persons may have 50,000 to 1 lakh kilocalories of energy stored as fat. The huge capacity of ATP formation by aerobic metabolism also relates to the nature of end products of these reactions. The end products of carbohydrates and fat catabolism are carbon dioxide and water. In addition to this, catabolism of proteins yields nitrogen containing end products easily. So they do not limit aerobic metabolism as lactic acid limits anaerobic glycolysis. Let us sum up our talk. Our body has a huge potential for ATP formation by aerobic metabolism, the equivalent of 2000 calories or much more. For comparison, recall that person referred to above as an equivalent of only about 15 kilocalories of CP in the body and a maximum of about 50 calories that can be derived from anaerobic glycolysis. I need to remind you here that aerobic metabolism can use either carbohydrates or fats as substrates. Proteins can also be used, but normally carbohydrates and fats make up nearly all the substrates during exercise. Maximum aerobic power is achieved when carbohydrates are the substrate for oxidative metabolism. The highest power with fats as substrates is only about 50% of the maximal power with carbohydrate as substrate.